I always love coming to the NFCR meetings. The diversity, the energy that Sudran brings is really makes this a very different kind of meeting. And thanks so much, Webb, for a great introduction. Most people actually appreciate that my most significant contribution to cancer immunology was marrying Suzanne Topalian. So as you heard, I am very much into one of the elements of cancer immunotherapy that involves blockade of these inhibitory signals T cells that we call checkpoints. Apology to the cell replication community for stealing their term. Alan DeAndrea still has not forgiven me for that. And there are many checkpoints and co-stimulatory signals that the immune system uses to regulate itself. The immune system is obviously very powerful, but it can also be very dangerous. So any system that is that powerful but also that dangerous needs lots of checks and balances to keep us from, for example, pushing nuclear buttons and blowing ourselves up. 3 of these checkpoints, blocking them, have now been FDA approved for treatment alone or in combination in various malignancies, and this is continues to be a growing and evolving area. As Webb mentioned, this is actually because people do ask me, they say, well, what actually have you done in checkpoint blockade other than being married to Suzanne Topalian? So yes, we did stumble on to PDL2. We originally called it B7DC. It's been renamed as PDL2. This is actually a duplication of PDL1. In fact, the 2 genes are within 60 kilobases of each other. They're the 2 most homologous of the B7 family members, and they both bind PD1. And with all due respect to folks who've developed anti-PD-L1 antibodies, as a class, it appears that the anti-PD-1 antibodies are somewhat more potent I think largely because they block the interaction of PD-1 with both PD-L1 and PD-L2. Although interestingly, after the genes duplicated, they modified their transcriptional regulatory regions quite a bit. And so they do have very different cell distributions, but PD-L2 is becoming more appreciated to also play a role in tumor immune resistance. So the very first clinical trials of these checkpoint inhibitors were done at a time when indeed there was a lot of appropriate skepticism about what the immune system could do in cancer. And this is Suzanne with, at the time, a newly minted assistant professor, Julie Bramer, in lung cancer, who together with a small biotech company called Medarex, we really worked very closely together to test panels of antibodies, of monoclonals that they were making, and ultimately bringing them into the clinic. And it was really an incredibly exciting time, and it's still been an incredibly exciting and wild ride, but it's gone in multiple directions. This is actually, we work with Medarex to develop the actually the first PD-1, PD-L1, and also LAG-3 antibodies after together, our group together with Dario Vignali, when he was actually at St. Jude, demonstrated that LAG-3 was a checkpoint. And this was actually the last meeting. We used to have these meetings twice a year before Medarex was about to become part of BMS, and that was certainly an exciting time that continued because their team remained as a part of BMS, and a lot of great stuff came out of it. Alan Corman is actually the person who physically made Opdivo and also relatlimab, which are combined now, the anti-PD-1, anti-LAG-3 in Opdualag. Nils Lundberg was the chief scientific officer, and again, really an exciting time. There are now 23 types of cancer, which in not only the advanced setting, but also moving to much earlier settings, and I'll talk a little bit about that PD-1 or anti-PD-1 or anti-PD-L1 blocking antibodies have been approved in. But I think the important thing are these percentages, and what you can see is that in the majority of cases, it's significantly less than 50% of the patients that actually derive benefit. There are two biomarkers, PD-L1 and also the genetic subset, which is mismatch repair deficiency or MSI, which are biomarkers. MSI is a fantastic biomarker to define patients, but it's only about 4 or 5% of patients. But all of those patients benefit tremendously from PD-1 blockade. PD-L1 is used in some cases. It's far from perfect, and Suja and I were talking yesterday, the need for better biomarker guidance to understand which patients are really going to benefit versus not continues to be very important. It's been great now to be recognized first in 2013. This is the first time that Science's Breakthrough of the Year didn't go to an investigator or to a particular discovery, but actually went to a field. Super exciting, and obviously the recognition of Jim Allison, Suku Hanjo for the 2018 Nobel Prizes really was, again, very exciting for the field, for our field, and sort of a coming of age. And one of the nice things about being in the Bloomberg Kimmel Institute is we get access to Bloomberg Intelligence. And so they actually made a graph of the annual revenues. So this is in millions. So this is now getting into the tens of billions. Just showing how immunotherapy relative to targeted therapy has come up in the world. And the point is not to say, it's actually getting to be relatively even, Steven, in terms of if you just look at revenues as one measure of impact on cancer patients. But the reality is that both the advantages in targeted therapy and immunotherapy and the combinations are truly an exciting dimension added to cancer therapeutics. This is looking specifically at some of the different checkpoints, and what we're going to start to see is that the proportion that are contributed by PD-1 is going to actually go down because others are now coming up. LAG-3 is coming up. This is ACT, which has been slow to translate into standard of care, but there are some real breakthroughs, including the first approval for an engineered adoptive cell therapy in synovial sarcoma. That's the first approval for solid malignancy. So that's sort of, I look at that a little bit as, breaking the Chuck Yeager, breaking the sound barrier, because people always said, will engineered ACT ever get out of liquid tumors? I always said, yes, it will. There's no reason biologically it shouldn't. And so now it has, and there's going to be a lot more there. Also to point out, and again, apologies for the mutations that occurred in going from Apple to Mac, or rather Apple to PC, but this is a proportion of PD-1 revenues that come from combinations with TKIs, and this is actually combinations with chemotherapy. These are going to grow. My prediction is that within a few years, there will be a very limited number of patients that out of the starting gate are treated with only an anti-PD-1. And so this, I think, again, is a very important area that needs to be attacked in a very thoughtful, scientifically driven and rational fashion, as opposed to just throwing stuff together, which sometimes can happen. So I'm going to talk about some of the science that we've done in the context of neoadjuvant therapy with anti-PD-1 and anti-PD-1 combination. So these are patients who are deemed to have operable disease, but based on the characteristics of their primary tumor, have a very high likelihood that they have micrometastatic disease, which after surgery, surgical resection of the primary, is ultimately going to relapse and kill them. And we've been able to learn a lot, not only about what's going on in the tumor microenvironment itself, but also to appreciate that part of what goes on, particularly with neoadjuvant therapy, that seeks to, for immunotherapy, to use the in-place tumor as a source of antigen to prime, ultimately responses that are being induced not only in the tumor, the primary tumor, but also in the tumor-draining lymph node, where blockade of the PD-1 pathway also plays a role. And the idea being that these lymphocytes in the tumor-draining lymph node will ultimately, when activated, leave the lymph node through the efferent lymphatics, go into the bloodstream, and then ultimately leave the bloodstream and essentially patrol through tissues for their sources of antigen, which if it's cancer, it's micrometastases. If it's a pathogenic infection, it's the same program that the immune system uses. This was the results of the first neoadjuvant anti-PD-1 trial that Patrick Ford led, and what you can see is, so this is essentially the percent regression. A lot of patients, this is lung cancer, and a lot of patients had very significant pathologic regressions, even though radiologically they still had tumor when the surgeon went in to do their resection. And then a lot of the tumors had minimal or very modest regression. But certainly this said that this was a very promising approach. Prior to this, neoadjuvant chemotherapy had been used to reduce the size of the tumor to make the surgery more feasible, more accessible to the surgeon. Here, the idea was to really ask, would this prime systemic anti-tumor immunity to be able to take out those micrometastases that the resection didn't get? And this is actually from the approval trial that Patrick Ford led, and also an analysis from Julie Stein-Deutsch, saying that the pathologic regression that you see when the tumor is removed correlates very highly with relapse-free survival or event-free survival, which basically tells us that by looking in the resected tumor, we can learn about elements of the immune response that are going to be relevant systemically. So I think this was a very important result showing that if you got a pathologic CR, this is with a combination of anti-PD-1 and chemotherapy, the vast majority of patients now going on beyond 3 years did not relapse. If you didn't get a pathologic CR, then the outcome was a lot worse. And this is an elegant study, one of two, this one from Sapna Patel, comparing neoadjuvant, adjuvant versus adjuvant alone. So adjuvant is you don't do anything before surgery, the surgeon removes the tumor, and then you treat the patient in their minimal residual or no evidence of disease NED state. And if indeed there is a priming effect of the neoadjuvant portion of the immunotherapy, these Kaplan-Meier curves should be the same. And as you can see, they're not. Also, the Nadina study led by Christian Blank showed very similar results, that you do better if you add neoadjuvant immunotherapy. And that, I think, is proof in patients that there really is the systemic priming effect, which is obviously very exciting. To the scientist, and this is just actually already the 6 FDA approvals for neoadjuvant immunotherapy. I also mentioned that neoadjuvant immunotherapy with anti-PD-1 for Merkel cell cancer, based on studies that Suzanne did with Paul Niem, is now in the NCCN guidelines. So this is really racing from the first publication in 2018 to now standard of care in more and more diseases. What this neoadjuvant approach also gives us is the opportunity to really dissect the tumor microenvironment. So this is, I love this sort of cartoon of the tumor microenvironment, because it really shows you how many cells are involved and how complex it is. And 15 years ago, we really could not have seriously contemplated doing the kind of deep dives into understanding these relationships that we can now. What I'm going to just talk about a little bit is one little snippet here, which are the cytotoxic CD8 cell, that's an end effector cell to kill the tumor cell, and also Treg cells. But to point out that this really is sort of looking at maybe the trunk or one of the legs of the elephant. Myeloid cells, and you'll hear about lots of different approaches in Lisa's upcoming section. Myeloid cells are critical. These unconventional T cells, even these gamma delta cells, are actually turning out to be very interesting. They're mate cells, but nobody here even knows what a mate cell is, maybe a few people. It's incredibly interesting and complicated, but now with various single cell high dimensional approaches as well as spatial approaches, we can actually wrap our heads around this and begin to understand the complexity of these interactions. I think we've probably at about the 2% level in terms of understanding these, but it's actually accessible. So we do a lot with single cell transcriptomics, and for those who aren't familiar with this, this is basically the TCGA. Okay, so you take a tumor, and Anna is smiling. TCGA has been, you take a tumor which has got all these different cells in it, and at the time there was no single cell transcriptomics, and you grind it all up, and you basically look at the transcripts of all the cells that were there. What single cell does is it allows you to basically do this, but even further, because if each one of these is a cell and these are groupings, you can now go in and profile each one of the seeds. These are the genes, because it's essentially every gene in the genome that is analyzed for every cell. So if you want to get into this, you definitely need some money. And you also need to become best friends with a really good computational biologist. And we were very fortunate early on to hook up with Hongkai Ji, who's just a fantastic guy and a wonderful computational biologist. And it took a while for the immunologists and the computational people to be able to start understanding each other's language. And it was great that this happened long before the pandemic, because we do these 4-hour dinner data meetings where it would sometimes take 30 minutes to go from one slide to the next so that we could understand what they were doing. So this led to some great work led by Kelly Smith, who started as a postdoctoral fellow with me and now is a superstar in the field, in which we did this single cell analysis of in the neoadjuvant studies, and combine that with an assay that she had developed called the MANIFEST assay, which stands for mutation-associated neoantigen functional expansion of T cells. That essentially told us specific T cell clones based on their T cell receptor sequence. And there are only 2 cell types in the body that carry essentially their own cellular barcode. Those are T cells and B cells, because of the uniqueness of their respectively T cell receptor and B cell receptor immunoglobulin rearrangements. We could then go in and use these to identify the small number of tumor-specific cells, which were identified by this MANIFEST assay, coupling this with single-cell coupled TCR-seq and RNA-seq to pick out in that ocean of individual TIL, the ones that were tumor-specific, pull out their T-cell receptors, do a transfer into a Jurkat line that was luciferase, in which luciferase would be activated when their T-cell receptors saw their antigen. And that essentially validated that the cells that we thought were tumor-specific really were recognizing peptides that contain mutations that we got from the genome sequencing or the exome sequencing of the tumor. And what this allowed us to do, and again, apologies for these mutations, is to do 2 things. One is to discover genes associated with dysfunction or reactivation after checkpoint blockade, because these were all being annotated based on whether there was a pathologic response to the anti-PD-1 or no response. So very simple dichotomous response, no response. What do you see in terms of genes that go up, genes that go down? Now not looking at all of the TIL, because most of them are bystander, but looking at that 1 to 5% that we validate as actually being tumor specific. The other thing that we found this could do is identify T-cell clones that were specific for oncogenic driver mutations. And this is going to actually come in terms of things going on now and into the future in terms of engineering. So once you identify these particular, we call them MANA-specific T cells for mutation-associated neoantigen, we can also do this for EBV-specific and flu-specific and any other virus that you want. And so you take your UMAP, which is this 2-dimensional reduction of that single-cell analysis. So each one of these dots is a cell. These are just the CD8 cells, and there are about 250,000 cells. It's actually a few bucks per cell. We use 10x as the platform. I learned that the reason that the company is called 10x is because to do these experiments costs 10 times what's in your budget. So what we can do is we can now this is actually an issue for young people who are actually trying to get into this field. These brilliant people who can't afford to do more than like one experiment, on their R01 grant. That's not a joke. So what we can do is we can now take the EBV specific, the flu specific, and the tumor neoantigen specific cells. So the neoantigen specific cells are in red, the flu specific are in blue, the EBV specific are purple, and superimpose them on this UMAP in which these different subclusters based on their transcriptional programs. And what you see is they're different. So the EBV specific cells are all in this effector cell cluster, whereas the flu-specific and tumor-specific cells are in a bunch of these clusters that are labeled TRM, which stands for tissue-resident memory. And you can see here that there are two canonical genes that define tissue-resident memory cells, CD103, which is an integrin, and the transcription factor Hobbit. I was very excited about this because I read the Lord of the Rings trilogy about 10 times when I was a kid, and I always wanted to work on Hobbit. And so essentially what this says is that the cells that actually recognize the tumor have been sitting in the tumor. They are actually resident in the tumor, but they're in a different tissue-resident memory population than the flu-specific cells. Remember, this is lung cancer. So these are all cells from patients who got the flu sometime in the past, and it's a respiratory infection. And so these are sort of the super memory cells that if they get hit with the flu again, bam, those cells will be activated really quickly. So we can now say, let's compare genes in the tumor-specific cells in the responders versus the non-responders. And a lot came out of this, but I'll just show you two examples that I think are very therapeutically relevant. One is that the level of IL-7 receptor, which is very important for homeostatic T cell expansion, maintenance of a memory population, is quite a bit higher than in the non-responders. Again, this is only looking at the tumor-specific cells. You could not see that if you looked at all of the TIL. Conversely, if you look at a checkpoint score, which are the 6 most commonly analyzed checkpoints, you find just the opposite, that the non-responders have much higher levels of checkpoints, again, supporting the notion of intelligent combinations of checkpoint inhibitors that may be able to improve significantly on just blockade of the PD-1 pathway. I'm actually going to skip this for the interest of time. So what I just described to you is extremely laborious and challenging. And so Hongkai, our wonderful computational collaborator, together with Zhen Zhang, they said, well, we should be able, just from the transcriptional profile of known validated tumor-specific cells, come up with an algorithm to be able to define which are tumor-specific cells without going through all the trouble of pulling out the T cell receptors, transferring them into these engineered Jurkats and all of the things that we've done. And so they developed actually a single patient linear regression voting model. This is an AI model based on 3 genes that were the most highly differentially expressed between tumors versus EBV and flu-specific cells. And those were CXCL13, this is CD8 cells, CXCL13, a chemokine, you can see almost not expressed in EBV and flu-specific cells, expressed at quite reasonable levels in MANA or neoantigen-specific cells, CD39, and then low expression levels overall of the IL-7 receptor. So much higher on EBV and particularly flu specific cells, much lower on tumor-specific cells. So they then used this. It's essentially a convolutional neural network where you're essentially iteratively training on ground truth validated T cell receptors, and came up with an algorithm that really just used these 3 genes, and then tested them on a set of independent data sets. And so these are the predicted tumor-specific cells. These actually, this is from a head and neck sample from Kai Wuker Fenwick's lab, and you can see where these were the cells that they had actually validated by T-cell receptor transfer. And these were against tumor-associated antigens, and you can see they match up very closely. This was a large data set from Steve Rosenberg's group for a number of different tumor types, looking at neoantigen, so mutation-specific T cells, and you can see they match up very closely. So we had the opportunity, this is a little bit wild, but to ask a really interesting question thanks to an NFCR grant. Thank you, Webb and the reviewers, to look at a very different kind of tumor antigen in Merkel cell carcinoma. And so Merkel cell carcinoma, it's a relatively infrequent skin tumor, but if it's stage 4, before anti-PD-1, the 5-year survival rate was about 10%. So this is really a bad disease, and 80% is caused by infection with a virus called a Merkel cell polyomavirus. 20% is caused by UV mutations. And it's really interesting because, as you might expect, the mutational burden of virus-negative Merkel cells, that 20%, is huge. It's like 1,000 exomic mutations per genome, whereas the virus positives have one of the very lowest total tumor mutational burden, on the order of 10. This is similar to pediatric leukemias that are driven by a particular oncogene. Yet, when you treat them with anti-PD-1, and this is actually the follow-up study, the original study that Suzanne did with Paul Niem being really one of the world's expert on Merkel cell. And the response is really quite impressive, 56% objective response rate, whether you're virus positive, which is the blue, or virus negative, which is the red. So what that says is that if you're virus positive, even though you've got hardly any mutations in your exome, anti-PD-1 will empower the immune system to take out that tumor. And it turns out that it's because there is a very robust response that's generated against that small Merkel cell polyomavirus T antigen, activated T antigen segment that's in the genome of these Merkel cells. And you can actually identify these cells with these peptide MHC tetramers. And so that allowed us to do the experiment of asking, could this MANA score, this purely computational score, how well would it do in identifying T cells specific, now not for a conventional mutation in the tumor, but actually for the Merkel cell polyomavirus. So on the single cell, one could superimpose this through this approach called CiteSeq, where you actually use these labeled tetramers that will pick out, they're the ground truth. They'll actually pick out the cells that are specific for Merkel cell polyomavirus T antigen. So those are these cells here, and then this is what Hong Kai and Zhen's MANA score picked out, and Tia here actually applied this and did all of this beautiful analysis. So the question is, how many of the validated tetramer positive cells did the MANA score miss? And again, this is a different kind of tumor antigen. And the answer is that of the 4,401 validated tetramer positive cells, the MANA score missed only 42 cells. There were an additional roughly equal number of cells that the MANA score said were positive, which could be because the tetramers don't pick up all of them. But what it clearly says is that for now, very different kinds of tumor antigens, simply computational, we can go into the single cell transcriptional analysis with a very, very high true positive rate and a very low false negative rate. So with that, we can now expand our analysis literally 100-fold with some of our own and other data sets. And so in fact, if you compare these putative T cell receptors picked out by putative tumor reactive cells picked out by the MANA score versus the non-tumor reactive cells, you can see that they have more of them have a tissue resident memory phenotype. Their checkpoints are higher. They're more cytotoxic, and they have a higher level of T cell receptor signaling. You'd kind of expect that because these are cells that are living in the tumor, and therefore their antigen is sitting right there. Now what we can do is we can actually look at the responders and non-responders now using this MANA score. So again, we've essentially with this, we can now increase our N by 100-fold. And some surprising things come out. So yes, again, the responders have a lower checkpoint score than the non-responders. But interestingly, the responders have a higher cytotoxicity score. And they also have a higher TCR signaling score. So the T cells specific for the tumor in non-responders are actually working harder to make the kinds of molecules that make them cytotoxic than actually the responders. Somewhat paradoxical. And if you look at the individual gene level, what you see is that indeed these are the genes in the non-responders, and you see granulysin, granzyme A, granzyme B. These are all of the cytotoxic granules that cytotoxic T cells try to use, and yet they're actually not succeeding in treating the tumor. Whereas if you look at the ones that are higher in the responders, you see IL-7 receptor, as we had seen initially. CXCL13 is higher, which means that if you are if you're an immune system in the tumor, successfully killing the tumor, you're making more CXCL13. But the one that always stands out, interestingly, is granzyme K. Granzyme K, it turns out, is not a cytotoxic granzyme. If you go back in the literature, I didn't know what granzyme K did. You can knock granzyme K out of a mouse, and it makes completely normal cytotoxic responses against viruses. What this is telling us is that these are genes that matter in successful fitness, and we actually have to understand why. Now I'm going to actually come close to the end. I'm going to skip a portion of it, but I'm going to now take us into the next depth of realm, which is spatial transcriptomics. So as if the dimensionality of conventional single cell was not enough, you can now essentially take that into spatial by essentially creating these small punch-outs in a row on essentially an FFPE section and begin to reveal some really interesting things. And so this is a case of a tumor that was deemed by the pathologist a non-responder because it didn't hit that conventional 90% decrease, or less than 10% of the viable cells were tumor. But when you look at these supposed non-responders, a lot of them have sections in the tumor that are just straight tumor nest, and you have other sections where you have very robust immune-mediated regression. The pathologist will say that we've got two different processes going on in the same tumor. They all get lumped together, and this gets called, formally it's read out as a non-responder pathologically, but the pathologist kept telling us it's more complicated than that. So now, and you can see exactly what they're talking about if it's not so obvious on the H&E. So this is EpCAM, which is a gene expressed in tumors. So now this is at the gene level. You can see that there are lots of tumor cells here in this tumor nest, and in the regression bed, there are very few tumors left. If you look at CD3, which is T cells, you can see T cells both in the regression bed and also in the tumor nest, even though there's no evidence that this tumor is actually regressing. There are T cells there, there are CD8 T cells there, and there are even Treg cells there, which I won't talk about in the interest of time. So now let's go back and look at granzyme B, which is the CTL granzyme, versus granzyme K, which our computational algorithms said are in tumor-reactive cells in responders. And you can see that most of the granzyme K are in the T cells that are in the regression bed, whereas there's much less granzyme K expressing T cells here. Now let's look at granzyme B. There are hardly any granzyme B expressing cells. There are some, but hardly any in the regression bed. There are a bunch of these granzyme B expressing cells in the tumor nest. They're just not doing anything. So our hypothesis is that granzyme K is marking a sustainable stem-like T cell necessary for anti-tumor immunity. This is actually spitting off killer cells. They do their thing and then they die. But if you don't have this sustained, again, this is a hypothesis that we obviously need to prove, this sustained progenitor population, that's really going to sustain within this tissue-resident tumor-specific memory population is going to keep generating ultimately the killer cells. So I'm going to skip over some stuff, and I just want to end. There's just so much great stuff. So one of the things that fell out of Kelly's analysis is that some of the tumor antigens, this will take 2 seconds, some of the tumor antigens were actually oncogenic mutations. So this is, for example, a P53 mutation, and this is one well from the manifest assay in which 4 clones popped out that recognized P53. So we were constantly in Bert Vogelstein's ear about this, and of course there was the MSI story, which was a brainstorming between the immunologists and the geneticists. And so ultimately, we managed to turn Bert Vogelstein into an immunologist. That's kind of scary. So Bert said, well, you know, we can target these oncogenic mutations with an antibody. I said, why would you use an antibody? You've got T cell receptors. Well, antibodies are a higher affinity, and we think we can do this. And in fact, they made a whole antibody library, and now this is a big collaboration. And the first one was an antibody that recognizes the most common P53 mutation peptide presented by the most common HLA allele A2. This was a great science paper, but it didn't really get its legs into the clinic until we were able to spin out. Originally it was called Manatee, now it's CLASP, and you're going to hear from Vipin Suri, the chief scientific officer. And what CLASP was able to do was to use combinations of structure-based affinity maturation, protein engineering, half-life extension, and ultimately, in record time, something we could never do within the university, take this to now impending an IND submission, which is actually going to be in a few weeks. So, so I think that NFCR's interest in really integrating the discovery science that goes on in academic institutions with the whole process of the public-private partnership and spin-outs really it makes a huge difference. Just to acknowledge some of the masterminds behind this, this happens to be actually a very strongly female-oriented. Most of the men, this is actually Arber's wedding, most of the men in this picture are actually the boyfriends or fiancés or husbands of the women that did the work. Kelly Smith is really the mastermind. Justina Kaushi, Jaja Zhang, Arber, and Laureen Chung really pulled off some of this amazing stuff. Particular thanks to Bloomberg Philanthropies, BMS, a great relationship that we had for a long time, Mark Foundation, and NFCR. Thank you.